So first of all, welcome everyone. I'm Maureen Clark, and we'll begin in a moment. Just before we start with a few of the little housekeeping notes, I want to first acknowledge the traditional owners of the country that we work and live on, their ongoing connections to the land, and we pay our respects to their culture and their elders, past and present. We recognize the traditional knowledges as well of the First Nations people of Australia in relation to the study and use sustainably of insects for food and medicine and other applications for thousands of years. So welcome everyone. Just a few little points. Please mute, of course, yourselves unless you're speaking to the group. There will be some natural areas throughout the presentation to pause, where we'll pause and ask some questions or see if anyone has any questions. Otherwise, you can use the chat, which some of us will be moderating, and we can have a little bit of Q&A at the end. And just in case I run out of time at the very end, I want to in advance thank Steve Moore for setting everything up this evening. Much appreciated for all the work behind the scenes. Thanks to Amy, of course, and thanks to Alex, who will facilitate. And for anyone who doesn't know Alex, let me just take a moment. Alex Arnold is the co-founder and CTO of Bardi and has over a decade of experience in developing black soldier fly larvae bioconversion technology and bringing products to market. Utilizing his background in genetics and agricultural science, he's delivered 2 industrial-scale BSFL facilities processing 15,000 tons per year of commercial food waste materials into animal feed and fertilizer for the Australian market. In 2022, Bardi launched Superfly, an organic certified 100% frass fertilizer and soil amendment product, which you can find in Bunnings stores across the country. And since then, Alex has developed a portfolio of BSFL-based fertilizer and plant biostimulant products, including formulations for major white labeling contracts, supplying the Australian horticultural industry. Alex has also led the development of live BSFL feed for large free-range egg farms, as well as establishing world-first carbon credit projects for BSFL bioconversion. So even a small fraction of all of this would explain why Alex is the perfect person to lead our discussion tonight with Dr. Amy Paten. So Alex, I'll hand over to you.
Thank you very much, Mo. Thank you for the lovely intro. Well, I'll be hosting this evening, and it is my pleasure to introduce tonight's speaker, Dr. Amy Paten. Dr. Amy Paten leads the Insects as Engineers team within the Environment Research Unit at CSIRO. Amy received a PhD from Massey University in animal science in 2015 with expertise in animal nutrition, functional genetics, and bioinformatics. Amy's research has spanned from large animal to insect production systems in Australia, China, and New Zealand. Joining CSIRO in 2016, Amy's research has focused on using systems biology approaches to understand and manipulate microbiome function and improved environmental outcomes and industrial applications. This has included projects on honeybee ecotoxicity, dung beetles for ecosystem engineering, and insect-based waste biotransformation. Current projects include work with the Victorian EPA on honeybee as biomonitors for PFAS, DevCom-funded research exploring the use of insect microbiomes for plastic biodegradation, and microbiome modulation and probiotic design to improve health and productivity of black soldier fly larvae. Tonight, Amy will share her insights from her work on microbiomes and probiotics in insect-based waste biotransformation and production systems. So it's an area that I'm deeply fascinated with, and I think has huge implications for the black soldier fly industry in Australia and globally. It's just so important if you think of the scale of a black soldier fly's gut, how important it is to, try and optimize that for either waste conversion or, productivity of the insect, or potentially, other wonderful products that we can make down the line. So as Mo said, please add any questions to the chat and we'll make sure we get to them at a suitable time. And without further ado, Dr. Amy Paten.
Thank you, everybody. Just check that everyone is seeing that screen. Oh, well, great. Excellent. All right. Thank you very much for that introduction, Alex. And yeah, thank you to Steve and Mo for hosting me as part of this AgriFutures Emerging Industries seminar series. So really excited to be talking about some of the microbiome work that we're doing at CSIRO. And thank you to everyone joining and giving up a bit of your evening to listen to me and engage in this talk. Just having trouble getting my thing working. There we go. Okay, for a little bit of context, I guess I just wanted to show a bit about where I sort of sit in CSIRO and maybe some of the larger capability that we have and what we're trying to develop in terms of waste biotransformation technologies. And so particularly in the organic waste streams, we're quite interested in developing different capability to be able to characterize those waste streams and then design specific solutions where we can valorize that waste into something beneficial. And that includes a lot of computational capability that's being developed to be able to kind of understand, predict, and design solutions for different waste streams, a lot of biotech development, including sort of enzyme development, but also microbial systems, which is largely where I sit, and also different kind of deployment platforms. And again, the black soldier fly system and the insect-based waste biotransformation is really where I sit in this kind of larger scheme. But this is just to show there's sort of an enormous amount of work happening at CSIRO and a whole lot of capability happening that I'm able to tap into as well for the work that I do. By microbiome, or maybe I'll start with black soldier fly. I probably don't need to explain to anybody here why black soldier fly are beneficial and why we love them so much in terms of waste biotransformation and insect protein production. They're obviously very good at consuming organic waste streams and converting that into protein, but also lots of other really useful byproducts as well, including frass, oils, all sorts of things. But like all industries, there can be challenges and opportunities, things like increasing the efficiency of bioconversion, being able to expand into maybe more difficult waste streams, mitigating contaminants that could be in organic waste, or targeting sort of different types of products, consistency, quality, those sorts of things. And so our research team is particularly interested in what role the microbiome plays in that and how we could use microbiome interventions to potentially address some of these industry challenges and opportunities. And by microbiome, I mean a community of microorganisms. So they're associated with a particular host or environment, and this can include bacteria, fungi, or archaea. Although for the purposes of this talk and most of our research, we really focus on bacteria, which is just kind of one component of the microbiome, but it's kind of by far the easiest to kind of characterize and manipulate. So that's where we focus most of our efforts. The microbiome, particularly in a host system, is shaped a lot by things like diet, host genetics, the environment, and the ecology of the host. And so I've just got 3 different kind of insect systems here that we've worked on to kind of show the differences in microbiome strategies that exist. So the honeybee on one end of the scale, they have a very consistent diet of pollen and nectar. And because of that, they've got a very strong co-evolved microbiome that's very highly conserved, very tightly regulated, and low complexity. They vertically transmit it through their trophylactic feeding from hive mates. And because of this, there's very little environmental influence on their microbiomes. They're quite stable. And from an intervention point of view, it's quite difficult to actually change their microbiome. At the next kind of level, you've got something like a dung beetle, where they have a much more complex and flexible microbiome. They do still have this kind of vertical transmission, in this case from mother to offspring, of what we would call a core microbiome, but they get some environmental transmission as well. And so because of that, there's sort of more flexibility in their microbiome and increasing complexity. Then you've got the black soldier fly at the other end of the spectrum, where they have a highly variable, highly complex microbiome. They don't have what we would call a core, so there isn't a set of taxa that we would always find in every black soldier fly. Varies quite a lot because they acquire their microbiome from the environment and largely from their diet. But there are certain functional genera that are frequently detected in black soldier fly. So I just want to highlight this study by Jdema et al. They did a meta-analysis of microbiome studies in 2022 and found these taxa are quite often found in black soldier fly microbiomes, but you don't see them every single time. And again, it really depends on their diet and the environment that they're in. But from an intervention point of view, because it's an environmentally acquired microbiome, it means we actually have a lot of scope to be able to introduce things into it and modulate it for specific purposes. In terms of characterizing an intervention, at CSIRO, we take what we would call a bottom-up and a top-down approach. So from a top-down approach, that's really where we're looking at these sort of more wild communities, much more kind of undefined. So if we were to take an insect gut and look at that, that's what we would call kind of an undefined community. And we'd need to use tools like sequencing and omics to actually then dig into what's in the microbiome, who's there, what they're doing, and what those kind of interactions are. And we can dig down and try and identify beneficial taxa or functions. From the bottom-up perspective, that's where we're taking individual isolates, so we know what they are. We can study them on a very individual level. We can also put them together in pairs or different combinations. And in terms of interventions, we can build those up into communities that we could deliver to the host organism for a specific function. So we call these syncoms. They're essentially synthetic communities. They're not necessarily genetically modified. It's just sort of an artificial assembly of individual isolates together to perform a specific function. And so these are kind of the approaches we use. And then in terms of interventions, we're mostly interested in probiotics, which is applying specific individual strains or consortia of beneficial microorganisms. But you also do have things like prebiotics, which is supplements that would stimulate growth of bacteria. And this is a little bit more non-specific. So you add things like fiber, like inulin, for example, to boost beneficial bacteria. The other alternative is postbiotics. So it's where you're looking at kind of the post-ferments or the metabolic byproducts of microbes. So there's not actually any viable live microorganisms, but there's things that they've produced that could be beneficial to then feed to a host. So I can pause here if there's any questions so far before I move on. But otherwise, I'll start going into a series of experiments we've done to sort of show some of the capability and what we're doing. But I'll pause for a minute if there are any questions.
Haven't seen anything in the chat yet.
Great. You're definitely my eyes and ears on this. I'll note with my shared screen, I can't see anything.
No problem.
All right. I'll move on to the next section, but feel free to interrupt if anything pops up. So this is sort of a study that we published recently. So this is by Helke Koch while he was working as a research fellow with us. And this was really just to kind of characterize not only the black soldier fly microbiome, but also microbiomes of native Australian soldier flies. So there's sort of thought to be up to about 300 species of soldier flies in Australia, although only half have been kind of characterized and identified. But we were quite interested in potentially accessing their microbiome richness from an intervention point of view. If there was something beneficial in native soldier flies that we could maybe apply to black soldier flies, that would be interesting to find. So we sort of wanted to have a look and basically characterize those microbiomes. We also were interested in the difference between black soldier flies in captivity versus wild. And by wild, I kind of just mean out there in compost bins doing their own thing, as opposed to be reared in the lab or in an industrial system. And so for this study, we sequenced the gut microbiomes of black soldier flies, so from 3 different wild source locations in the ACT, Queensland, and Northern Territory, as well as the ones that we've reared in the lab. And we also sequenced the gut microbiomes of 4 different native Australian soldier fly species that all have slightly different diets and ecologies. And what we found was quite interesting. So we did obviously see quite a lot of variation in the microbiomes. Again, that's just because they're largely driven by environmental differences. And so you can look at a species like Inopus, which is this guy up here, that's the adult. This is the larvae. They're a pest of the sugarcane industry. They feed on the roots. So it's quite a specific and narrow diet. And for this reason, they've also got quite a specific and narrow microbiome compared to the other soldier fly species that obviously feed on more heterogeneous organic matter, and black soldier fly obviously feeding in waste streams. We do see some conserved taxa across the different soldier flies. So there are clearly certain taxa that are beneficial for feeding on organic matter, and those are ones that we're potentially interested in that we want to make sure we have in our black soldier fly. But one thing we noticed that was quite interesting is when we looked at our lab line. So in particular, compared with the wild black soldier fly, you might notice our lab line has a really depauperate microbiome in terms of ASVs and also in species richness. And this is not something that's uncommon for captive bred animals because of the way they're reared and because of the lack of diversity in their diet. And our lab line are just fed chicken feed all the time. They get this real reduction in their microbiome. And that's just something to kind of think about in a rearing kind of system. If you have them on a consistent diet, how they can lose some of that diversity, and then what that might mean if you are suddenly switching up their diet or wanting to introduce new functions. Again, happy to pause if there are any questions about that study. It is published as well, so people, if they want a bit more info, are welcome to have a look and read that study as well. If there's no questions, I'll move on. In this study,
Amy?
Oh, yes, happy to go back.
Sorry, David Allen has a question. David,
Great.
do you want to go ahead?
Hi, Amy. So I think if I understand the implications of this, the lab-reared insects or larvae really have does this mean they have a lower chance of being able to digest a wider stream of waste material? So, for example, if rather than a consistent side stream, you're feeding them very mixed waste and variable waste, the wild black soldier fly will in fact do better generally. Is that likely?
Yeah, that's kind of the assumption. And we do have a study a bit further on where we look at this a little bit more. So we'll go into that a bit more. But yes, that's kind of generally the assumption, is that you'll get better performance with a more diverse microbiome, just because you've built in a bit more of those kind of metabolic functions. You've got a bit of redundancy as well in what they can do, and it's a bit more flexible and adaptable. There are also implications as well for things like gut dysbiosis and suppressing pathogens too. So there's sort of the ability to be able to break down more diverse waste streams. And then there's also sort of general health and pathogen suppression kind of implications as well. I mean, black soldier fly do have the ability to kind of change and have a flexible microbiome, and it can be rescued, which again, I will get to in a next study. But yeah, generally what you want to see is a nice, rich, diverse microbiome like this. It's not just for black soldier flies, it's true for most animals. If you have that kind of diversity, you've just got a lot more kind of flexibility and adaptability in terms of those metabolic functions and substrates that you're able to digest.
Excellent. And I don't think many of us would have had the chance to have the bottom right diagram explained previously. So could you just give us a bit of a picture of what we can see in each of those quadrants, and why does the lab one look so blank? Excuse my hiccups.
Yeah, that's right. This one here with the box around it.
Correct.
Yes. Yeah, so this is a heat map. And so basically we've got different genera down the side of bacteria. And if there is a dot, it means it's present, and the intensity of that dot is the abundance. So the really dark colored dots mean that's in really high abundance. So what we see in the black soldier fly is that there are some really highly abundant taxa, but that diversity is there. So there's a lot of taxa here where it's white, where they're not present, where we see them in the wild black soldier fly. And we know a lot of these can be really beneficial as well to black soldier fly. So for us, it was a bit upsetting that they weren't in our lab line. And probably look to try and introduce them. And it was also, so if you look across as well, basically if there's a line, it means it's present in all the different samples. So you can see where there are certain taxa where they're kind of conserved and present across. And then you can also see, obviously, where there's taxa missing, where they might be present in some of the other samples. And so for us, I guess we're interested in maybe tapping into some of this diversity and seeing if it's possible to perhaps, take this and introduce it back into our lab lines and kind of restore their microbiome function. And we're also interested in understanding, again, what this means. So for this study, we really just characterized the microbiome. We didn't follow up with any kind of feeding trials or phenotype analysis or anything like that. This was really just to kind of have a look and see what microbiomes look like, how much diversity there is, and that kind of influence of being in the lab versus being able to feed on a quite diverse diet of whatever's in somebody's compost bin.
Excellent, thanks. And how many generations are your lab-reared larvae?
Yeah, these ones have been in the lab for a very long time. So this is from our Canberra lab, but we got them originally also from the Brisbane lab. And so as far as I know, it's been sort of maybe almost 10 years of being in the lab. So yeah, a long time.
Thank you, Amy. How about a couple more questions from the chat? Martin Pike has asked, is this only from one generation of larvae or across multiple generations that we see the biome changing?
Yeah, it's a great question. So in this study, this is just one generation. I will point out the Exarita strains. So the Exarita was wild, and we also started to rear them in the lab. In this instance, they'd only been in the lab for like 1 or 2 generations. And you can see they're starting to lose some of the diversity in their microbiome, but definitely not to the extent the black soldier fly ones have. So it does take a little bit of time for them to start to lose that diversity. And again, it's really just because their microbiome is so linked to their diet mostly. And so if they have taxa that aren't really that useful for the diet that they're on, or other taxa that just do really well on that diet and kind of can outcompete, they start to dominate the microbiome. And the next study I'm going to show too will show a little bit about how kind of rapidly that microbiome changes. And there was one study, which I'll talk about at the end, where we started to look at directed evolution, but in that case, we just did it over 2 generations. But again, starting to look at how it changes over generations. We haven't actually done a big multi-generational study yet. That's definitely kind of on the cards. And I think when I get to the last study with the directed evolution, that's definitely where we want to take that one, to start to really dig into how long kind of does it take a microbiome to stabilize and start to kind of persist over generations.
And if it's time for just one more question on this section, Phoebe had a question, which I was also wondering, is what were the lab-fed larvae reared on? Were any fed a variety of food waste or a TMR as opposed to just chicken feed?
Yeah, the lab ones are just reared on a chicken feed diet. So the only time we ever put them on other diets is when we're running experiments. So the larvae are taken and put on an experimental diet. But like the general strain that we just maintain and rear in the lab, they just receive a chicken feed diet.
Okay. Cool, thank you.
That's right. It certainly obviously raises implications too for people studying black soldier flies in a lab system on a lab diet, and how much that can potentially apply to industry. So it was definitely a good finding for us to kind of see and be aware of.
Yeah, I think in some settings, the lab setting or the industry setting, it is quite a diverse food waste stream. So do you think if in a lab setting or industry setting, if the food waste stream is really diverse, so from commercial or domestic food waste or agricultural waste streams, that you would see the biodiversity of the microbiome of that lab strain of BSF? Would you see that microbiome diversity, replicate what you're seeing, or even possibly exceed what you're seeing in the wild microbiomes?
Yeah, I would expect if you have an industry setting or a lab setting where you are feeding them a diverse diet, that yes, they should have a more diverse microbiome. It's not something we've tested yet with our lab line. We're sort of thinking more along the lines of just trying to introduce more microbiome diversity back into them. But yeah, certainly thinking about switching up the diet and how we could maintain diversity is also important. For the most part, we kind of keep our lab line. And then if we are trying to do industry trials, we've been really lucky with support from industry in terms of getting their actual industrial lines of larvae to try for the experiment. So that's a bit more relevant as well. But yeah, I think their diet really does make a difference. So I would expect if they're getting a very diverse waste stream diet, they're likely to have a more diverse microbiome. And then obviously if they have a more kind of narrow diet, that microbiome is going to sort of start to converge on the same thing and sort of maintain.
Yep, cool. Thank you. Makes sense.
No problems. So for this study, and this is one where we did use an industrial line as well as our lab adapted line, but this was part of Sophia's postdoc, and she was interested in incorporating wastewater biosolids into black soldier fly larvae diets. And she was interested in that question of how does the microbiome adapt? Do you need to, is it better to give them biosolids from day 0 so that their microbiome can kind of establish on biosolids and that's the microbiome that they have and they'll do better on biosolids when they're a bit older, a bit later on? Or does the kind of normal system where you kind of have them on a neonate diet and then you switch them onto a waste stream later on work well, and will that microbiome adapt? And so she did that here with a control diet. So they had a hatch diet and then a mixed vegetable diet as their diet from day 6. The biosolids treatment, so they received 50% biosolids, 50% hatch diet from the neonate period, and then was switched to vegetables, but still with 50% biosolids. So they had biosolid exposure all the way through. And then the diet switching treatment, which is where they had the larval hatch, but no biosolids, and they only received biosolids in their diet at day 6. And she looked at a whole bunch of larval phenotypes, and then also at their microbiome. So the diet switch happened at day 6, and then day 9, day 11, and day 13 were days after that diet switch had occurred. And one thing that we found, which was quite interesting, was that the microbiome does establish quite early, and it is very influenced by diet composition. So if we look down here, this is the beta diversity of the microbiome. So this is showing the different samples, green ones being the larvae that were fed biosolids diet all the way through. Ones that were purple color are our controls, so they've received the vegetables, and then the ones in orange are the ones that were initially fed the larval hatch diet, but then were switched diets onto a biosolids diet, so they only received biosolids from day 6. And then in this last plot, we've also got the microbiome of the actual biosolid substrate itself. And so what we see at day 6 is the biosolids, the microbiome and the larvae-fed biosolids have a very distinct and different microbiome from the other 2 treatments, which are on the same diet at this stage. But then once that diet switch happens, we do start to see a shift. Interestingly, the biosolids larvae, their microbiome, it's established pretty early and it stays the same through the whole time point. It doesn't really change. In the controls, we actually see a bit of shift in the microbiome over time, but it always remains different from the biosolids diet. And we think this is potentially because, again, that vegetable diet's a little bit more mixed, a little bit less homogenized than the biosolids, and the larvae are sort of able to change and adjust their consumption for their developmental needs as well, and have a bit more kind of, heterogeneous consumption of vegetables in that diet. And then what we do see, which is really nice, is the ones that had the diet switch, where their microbiome was very similar to the controls, over time started to shift, and it started to shift away from that control microbiome and shift towards that biosolids diet microbiome. So by day 13, they effectively had that microbiome now adapted. But yeah, over time, the microbiome was changing. And the microbiome of the biosolids fed larvae was quite distinct from the actual biosolid substrate. So even though biosolids are quite rich in microbes, it doesn't necessarily translate to exactly the same microbiome. So there's some selection in what microbes actually establish in the larval gut from the environment and from the diet in this case. One of the things that was really interesting, if we're looking at the alpha diversity, is how the lab adapted and the industrial line performed. And so one of the things we noticed, particularly with this diet switching treatment, was that our lab adapted line had really low survival. So they actually were fine if they fed biosolids all the way through, and they were fine if they had a control diet all the way through. But when we did that diet switch, our lab adapted line really dropped in larval survival, whereas the industrial line was fine with survival. And when we look at that microbiome, what we see is in the industrial line, that shift in microbiome. So again, the colors are the same, the middle one being the diet switch, bottom being the biosolids diet, the control diet. The colors are a little bit mixed up. Yeah, the biosolids diet at the top, the control diet at the bottom. What we see is quite a rapid shift. So by day 9, that microbiome is almost up there with the biosolids diet, whereas in our lab adapted line, we see a much slower shift in that adaptation. They still do adapt, but it's happening later. And so we think that potentially has implications for how they're able to survive on that diet. So that's quite interesting. Again, we think because the industrial line, the way they're reared, they are reared on a hatch diet and then transferred to a food waste stream, whereas our lab line just fed chicken feed all the time and they're not really used to a diet switch. And so we think potentially the larvae and the initial microbiome they had was maybe not as well adapted to that kind of setup as the industrial line. So again, just an interesting take home about kind of priming microbiomes. If you are wanting to make a big switch in their diet, potentially exposing them earlier can help prime that microbiome. Alternatively, and what we're interested in is identifying what that microbiome is, and you could inoculate that in early and basically have them kind of pre-prepared for a diet that they move on to. So they're effectively already at this point where they've got a microbiome that's good to go. But yeah, and again, just a point about studies in labs and how they apply to kind of real world and industrial scenarios. And again, happy to take questions at this one.
Thank you. I have a couple of questions from the chat. David Allen has asked, will the diversity redevelop if the lab larvae return to, for example, a very diverse range of waste and fermented products?
Yeah, I mean, this would suggest so. Yeah. Sorry.
Oh, I think you've answered that.
Great. Yeah, I mean, this would suggest so. I think this suggests they do adapt, but yeah, it'd probably be something we need to just do a bit more to keep it there.
And David's also asked, by bio-waste, are you referring to human waste, or is it perhaps like agricultural bio-waste? So
no, this is from human bio-waste, so from wastewater treatment plant.
Okay.
That's the picture of it there. It looks quite kind of soil-like. Yeah.
Yep.
It doesn't smell great.
Thank you. Anyone have any other questions?
I'm happy to move on if we're ready.
Yeah, sounds good.
Excellent.
Looks like, sorry, this is Steve here. Looks like we do have another question down there from 6:34 p.m. Yeah.
Oh, sorry. Yeah, it just popped up then. Will the microbiome inside the gut differ according to the waste that it's fed?
Yeah, it should. That's what most of these studies have kind of indicated, is that once you change the diet, that microbiome starts to shift. So yeah, this middle treatment, this orange one in particular, we're showing there was a starting microbiome, and now that they've changed diets, that microbiome has shifted quite a lot, and it's different from the one that was just kept on vegetables all the way through.
I have a quick question, and that's what were the major outcomes of being able to adapt more quickly? So the industrial strain adapted more quickly in its response to the particularly to the mixed diet, and you saw Did you see improvements in the performance of that as a consequence?
Yeah, I mean, so the biggest consequence was really in survival. So we actually had quite high mortality in the lab adapted line versus the industrial one, where there were no kind of mortalities that happened, and they were basically the same as the other treatments. And we really only saw those mortalities in the diet switch. So the lab line fed biosolids all the way through, survived fine. Because the survival changed, it meant in our little trays, the stocking rate effectively changed. So it sort of
Hmm.
started to skew some of the other data
Yeah.
then around, consumption. I think, as you know, if they've got a bit more room to move, they do start consuming more food. But yeah, the biggest thing was the survival.
Okay. And do you have any insights perhaps into how how you could further optimize how rapidly an industrial line could adapt to a diet switch? So it might be, quite standard in industry to rear neonates
Yes.
on chicken feed or Gainesville or whatever it is, and then switch it
Yeah.
over to what the actual waste that you're processing is. So how could we further optimize that adaptation so we don't get a little bit of a dip in that swap?
Yeah. Yeah, great question. And I mean, that's something obviously we're very, very interested in. And from our perspective, we're hoping that a probiotic supplement would be able to do that. So in this case, feeding them biosolids was able to kind of prime their microbiome, but you don't really want to do that because your industrial system has your neonate stage and then your waste stream stage, and potentially your waste streams are different as well, not always the same. And so what we would like to do, or what our kind of vision is to be able to come up with probiotics that are kind of tailored to the waste stream that the larvae are going to be moved on to. So if you're moving them on to a biosolids diet, you would have a microbiome that's very efficient. It would help the larvae break down that biosolids diet. If you're moving them on to a high cellulose diet, you could have a microbiome that helps with cellulose. If you're moving them onto a high protein diet or a meat-based diet, you could have a microbiome that you basically just inoculate in at the larval stage, at the neonate stage, and then that would basically set them up for then once they're moved on to a waste stream where they're kind of primed and ready to go. We've done a few initial trials with this, and we have seen some differences, but still very much a work in progress, but that's kind of, I guess, the vision we're working towards, is to be able to have those sort of tailored probiotics, or even just a general probiotics that gives them that really rich microbiome diversity and flexibility that means they're going to be good regardless of what diet you put them on. And so those are the things we're kind of interested in and we're currently kind of testing and working on at the moment.
Yeah, nice. Thank you. And then David's also asked, some have sampled the BSFL gut and shown microbiome variants in different sections. Is that studied? Sort of for
Yes.
that mid-gut. Yep.
Yes, that is one of the results I'm going to show.
Great. All right. I'll let you crack on then.
Great question. So this is before we get to the gut segments, but this is kind of one of our first intervention studies. So maybe revisiting that first study where I showed our lab line had a very kind of depauperate microbiome, and we were interested in, could we rescue that? Could we restore functions by basically doing a microbiome transplant? So kind of the equivalent of like a fecal transplant that you would do in humans, but in this case, crushed up larval guts from another source. And in this case, we're also kind of interested in how far we could push that. So could we actually do a cross-species microbiome inoculation from, in this case, a closely related soldier fly species. So this was the Axarita spinigera, which is one of the native Australian soldier flies. And they feed on a diverse diet of decaying organic matter. So probably the most closest to a black soldier fly diet. And we thought kind of more likely to have a beneficial microbiome that we could potentially transplant into our lab line that were lacking in their microbiome. This was done in a very controlled azenic gnotobiotic system. So for this study, we surface sterilized the eggs and incubated them on sterile agar and transferred them onto a sterile diet. And so this means they basically don't have a microbiome until we actually inoculate them with a bacterial suspension. And in this study, we inoculated them either back with their original lab black soldier fly microbiome, or we gave them the Exarita microbiome, and we also gave them a combination of both the Exarita and the lab black soldier fly microbiome to see how those two microbiomes would interact. And then we also did have a sterile control. And again, this was just done on a chicken feed diet, which is their usual diet. We saw some pretty striking results that were a bit of a surprise for us, but really interesting. And so these photos are all taken at the same time point. And what we see here is the azenic ones. So these are the controls that didn't receive any microbiome. And you can see they're very small and sad and unhappy without their microbiome. These are the ones that receive the lab black soldier fly microbiome. So they do have a microbiome, but it is that kind of more depauperate lab microbiome. And obviously they're growing better and doing okay. But when you compare them to the black soldier fly that received this transplant of microbiome from Exarita, either by itself or in combination with the black soldier fly microbiome, you can see that they're grown bigger, the development is increased, and yeah, just doing a lot better in terms of the weight and the survival as well. So really, really marked improvement just by, adding in that extra microbiome diversity to them. And when we sequenced that microbiome diversity to see what actually was able to transplant across, what we found was that a lot more diversity was able to be introduced from the Exarita. So in this block here, again, we've got the heat map. So with the squares showing the presence and the darker intensity being a higher relative abundance, a lighter color being a lower relative abundance. And we've got all of our taxa down here. And we've got our lab line here. You can see lacking in a lot, but now rescued by the addition of the Exarita microbiome. And that Exarita microbiome, even when in combination with the black soldier fly microbiome, really kind of dominates. So it was interesting. We do see some taxa that only established in the Exarita only microbiome transplant, so obviously didn't compete particularly well once the black soldier fly microbiome was also present. But we did see, a lot of commonality. And we also saw a lot of genera come back, Disconomonas, Scrophomicrobium, Citrobacter, that were absent in our lab line that are known to be really beneficial to black soldier fly, particularly in terms of, breaking down kind of more complex materials. One thing we did notice as well in our lab line was a really high abundance of Pseudomonas, which is just here. And that's relevant because Pseudomonas can sometimes be a pathogen. And in this instance, we noticed a single Pseudomonas aeruginosa ASV was notably abundant in all of our black soldier fly inoculated samples. And so this is them here. This is actually in the diet, the frass, and the larvae. So it was sort of present in that whole system. And what we noticed was in the black soldier fly plus Exarita combination, it was there, it was still present. So it did successfully inoculate across from the black soldier fly component of the microbiome. But having that Exarita component of the microbiome really suppressed the establishment and abundance of this strain. And that's relevant because in this case the strain was pathogenic, so we did isolate it, we did sequence it, and we did find it was one of the pathogenic strains, did have all of the virulence factor genes that you would expect. And when we did do a trial where we inoculated this strain into our azenic system, any of the larvae that had this strain had really low survival compared to those azenic controls that didn't have any microbiome. So it really does have an effect on their survival, which, going back to those original kind of differences in the results, is potentially one of the kind of explaining factors. But also just like a really good example of another benefit, I guess, of having that diverse microbiome is that it can offer that kind of protection against pathogens and really boost health in black soldier fly larvae. Happy to stop for questions on that one.
Thank you. I have a
David Allen,
oh, yep. Sorry.
I was going to say David Allen's hand is still up. Is that for a question or?
Sorry, David. Go ahead.
You go, Alex, please.
Oh,
Done.
Thanks. I was just going to ask. Following that, it looks like one thing that wasn't done, I guess, was testing the industrial guts, inoculating an industrial black soldier fly gut into a neonate. So, do you think it would be practical to inoculate a neonate's gut by, essentially crushing up some BSFL from the industry strain, whatever that's currently being used in the industry that's been fed on mixed food waste, and just take that mush and put it into the neonate food? Or is there is there a more optimal way to inoculate the guts of the microbes of the neonates that perhaps doesn't introduce pathogens? Or how can we get the best of both worlds?
Yeah, I mean, so I guess this is like an example of what we call a top-down approach, which is where you're putting that uncharacterized microbiome in as a supplement. Like I said, kind of similar to the human equivalent of the fecal transplant. And yeah, the risk with that is you don't know what you're introducing, and you don't know if you're introducing a pathogen. And so that's why I guess a lot of our work is focused much more around that bottom-up approach for an actual kind of more commercial application where you have known strains that you've assembled together in a beneficial community, so you know what you're introducing and you know that there's no pathogens in there. But if you're just sort of in a industrial setting and you're wanting to kind of make sure you have microbiome that's transferring across generations, just transferring the frass can be a good way of doing that. So one of the studies later down where I'm looking at the gut segments, we also have the frass, and you can see the frass has quite a similar microbiome to the gut microbiome as well. And certainly for us with our lab line, that's something we're thinking about doing, is just enriching them with certain isolates that we've got that we know are beneficial and that we know are missing from them. So things like Disconomonas, Microbiome, we have isolates of all of these. So we can assemble them together and make a little probiotic to put that in with our larvae. And then to make sure it stays in the system, we probably just transfer a bit of the frass and residue each time to the next generation to make sure they continue to pick up those beneficial microbes. So that's sort of one way of doing it. But if you do want to kind of intervene with more precision, definitely that kind of more bottom-up approach where you have specific strains that are together, that you know what they are and that you want to be in your microbiome, is what you would deliver.
Yep. Cool. Thank you.
Oh, good. Are there any other questions? Yep. That's all. Great.
I think that's all for now. Thank you.
That's okay. Thanks for that. So this is the study where we really wanted to dig in to what's going on in the gut segments. So again, this is another diet mixing study, again, just because that's something that is commonly done in industry. And we sort of had interests around what kind of effect and role the microbiome has in that. And we also wanted to use this study for like a really in-depth characterization to get good spatial resolution of the microbiome, but also really good functional resolution, because most of the studies we've done so far have just been sequence-based. So we've been looking at the taxa that are there, but we haven't necessarily been looking at the functions they're doing. So for this study, we had an oat milk waste that we were provided from industry, and we had our normal lab diet, which is chicken feed, and then we also did a combination, so just a 1-to-1 wet weight of chicken feed and oat milk waste as a diet. And we did a starter diet, and then by day 5, we switched them over to the treatment diets. And in this study, we sampled them after 12 days, and we looked at some phenotype, but we also dissected the gut into different segments. So these are different segments of the gut that are largely separated by kind of the pH and the conditions in the gut. And so you've got the anterior midgut up front, you've got the middle midgut, the posterior midgut, and then finally the hindgut. And for this, we wanted to be able to characterize the microbiome structure in each of the gut segments and also the function. And because we were getting genomes for everything that was in each of the gut segments, we also wanted to build what we call genome scale metabolic models, which are essentially a mathematical representation of all of the metabolism that an organism can do. And it's based on a known set of inputs. And then you can see all of the reactions and metabolism that happens to produce a biomass output. But for this model, because we have all of the different species happening in each of the gut sections, we're able to do a community model and look at how within a gut segment they interact with each other and where there's cross-feeding. And then we're also able to do a whole digestive tract model as well, where the outputs from one segment of the gut become the inputs of the next segment of the gut, and so on and so forth. The only thing we haven't done at this stage is incorporate the host into it. Modeling eukaryotes or higher order organisms is very, very difficult in terms of doing a genome scale metabolic model. There is one for black soldier fly larvae. It's not the best though. So at the moment, we've really just focused on kind of modeling the microbiome and what's going on there, and kind of assuming that things are being taken up by the host. But eventually one day, I think we would like to also try and get a host microbiome model as well. So for this study, one of the things we did see in terms of phenotype was that the combined diet had a lot better growth than the other two treatments. That wasn't really a surprise in terms of it doing better, just because you've kind of got a balance of a carbohydrate-rich chicken feed and then more of a protein-rich oat milk-based diet. But we were a little bit surprised just by how much better they grew. And again, we really wanted to dig into the microbiome and work out, was there anything beyond just the nutrients balancing? Was there something in terms of the microbiome, like were there microbes that were present in this diet and microbes that were present in this diet that when you put them together, they were really synergistic and really helped? Or was there something emergent? So that's kind of the questions we were interested in. And then in terms of characterizing, as I mentioned, we used genomics to sequence the gut, but also proteomics to have a look at the proteins being expressed. So this is actually what those microbes are doing in the different segments of the gut. So here I've got a bit of a summary of some of the metagenomic results and the microbiome composition in the different diets, and also comparisons between them. This is the 16S, the full-length 16S sequencing down here. And so we've got the chicken feed in the first box, the oat milk waste in the last box, and the combination diet in the middle box. And we've got down the axis here, the first one is the actual diet substrate, so before it was fed to them. Then we've got the different gut segments, so the mid-gut segments, the hind-gut segment. And then we also have the frass as well, the residue that was collected at the end. And so in terms of just looking at the microbiome structure, it's really obvious to see the chicken feed is quite different from the other two, and again, a little bit less diverse. Whereas once we add the oat milk waste in, we start to see a bit more of a diverse microbiome, and the oat milk waste also really drives kind of most of the microbiome composition that we see in this mixed diet. But we do also see some novel emergent taxa that we didn't see in either of these two diets. So particularly up here, we've got Anaerocolumna and Anaerosporobacter. So we actually see particularly a lot more anaerobes and fermenters that are coming out when we have this combined diet that we didn't see in the individual diets. We do also see most of the differences tend to be in these mid-gut sections. That's where there's a lot more kind of diversity compared to the other treatments. Whereas in the hindgut, it's largely dominated by Providencia retgeri. And that is also just a very dominant species that we see in black soldier fly microbiomes. And you can see this beige blob here. It's really taking over a lot of the microbiome. In terms of actual kind of comparison, so we wanted to see what was specifically different between the chicken feed and the combined diet, or the oat milk and the combined diet, which is this plot over here. So again, we've got the diet, the different gut sections, and the frass. And this is a contrast, so it's the combined treatment minus the chicken feed, with the colors being an up or down regulation. So the darker the color, the higher the increase or the decrease, and the circle size is kind of the log fold change. So a bigger circle is a bigger fold change difference in that abundance. And here we've got the different taxa. And so again, it's quite noticeable. Most of the differences are between the chicken feed and the combined diet, which is what we expect from the 16S. And we do see the differences are generally quite consistent in the different gut segments. There are a few instances, though, where we see a difference only in one specific gut segment. And so that's kind of quite interesting. And we do also see in the FRAS, like a lot of the FRAS is quite representative as well of the gut differences, whereas the diet, the initial diet itself, didn't actually have much of a microbiome in it to begin with. And it's also just not even remotely predictive of what the gut microbiome would look like. But by changing the diets, you can change the gut microbiome, and you can also quite reliably change the FRAS microbiome, which is interesting if you are interested in the FRAS and the microbial content of the FRAS. If you're using that for like a soil amendment or something and you wanted specific bacteria to be in there, you could use a diet intervention or a probiotic intervention to achieve that. In terms of the metaproteomics, again, it was largely reflective of the metagenomics, and sort of unsurprisingly, this Providencia rectory that really dominates the microbiome is really also dominant in the proteome in terms of protein abundance. So that's not a surprise. It's there in high abundance. It's doing a lot of things. So we expect that. What we did find quite interesting was some taxa. So this is Klebsiella oxycoca. It was really, really rare in the metagenome, but actually one of the most abundant species in the proteome. And so even though it's a really rare taxa, it's actually doing a lot. There's a lot of function happening. And so that can be also really important, because I think a lot of time people often just do sequence-based studies, not necessarily dig into proteome or metabolome or functional, actual functional realized differences. And they might be missing something that actually could be really beneficial and useful. And so this is one strain that we're kind of looking into for probiotic development because of some of the beneficial functions it can have. And yeah, again, very similar to the metagenome in that most of the differences are really between the chicken feed and the combined treatment, with actually very few functional differences between the chicken feed and the oat milk waste diet. And this one shows also the gut segments that the differences are in. So again, quite reflective of the metagenome, and that it's the midgut that is where most of the differences are. And so from an intervention point of view or probiotic design, that's really important for us to know, because we want to be picking taxa probably that are here in the midgut. That's where we're probably going to be able to change things. And you also want to know where things are co-located. So if you want them to be kind of working synergistically together, you need to know if they're co-located, and you also kind of need to know the order that metabolism happens. If you have something breaking down a substrate and then another strain that breaks down the output of that, you need to also, again, make sure it's located in those sequential parts of the gut. So in terms of like probiotic design and syncom design, this is really useful information for us to be able to get that really good understanding of who's in the gut, where they are, what they're doing, and who they're potentially interacting with. As I said, the next steps for this are that metabolic model. So we do have that running, and there are some really interesting results, but we're in the process of writing that up as a paper. So I'm unable to share those right now, but definitely a watch this space, I guess, and hopefully we'll have the paper out this year. Yeah, happy to take questions on that one.
Great. We have a few questions. David's asked about the oat waste. So by oat waste, do you mean husks and bran, etc., or oat straw? And then following up, if you were to be designing a microbiome suited for high performance on oat straw, wheat straw, or rice straw, how would you do it?
Yeah, so this was oat milk waste from a company. So it was kind of like the sludge that's left with the liquid removed mostly. So yeah, it would be kind of the husk and the bran, that kind of thing. Yeah, so not just sort of oat, bit different from oat grain, because it's obviously been through a bit of processing and treatment, and it had sort of fermented a bit as well. So a bit different from maybe like a raw grain processing, but yeah, it was quite high. We did a nutritional content analysis. Yeah. Yeah, it's like when you get oat milk and you get the dregs at the bottom. But yeah, it was quite high in protein. It was very high in protein.
Yeah. And he had a follow-up question. If you were to be designing a microbiome suited for high performance on oat straw, wheat straw, or rice straw, how would you do it?
Yeah, I mean, so that's, I guess, where we're hoping to go with this. This, again, top-down approach where we are looking at the microbiome and trying to drill down into what are useful taxa and what their useful functions are. And that's sort of where we've got to with the modeling now, where we've been able to identify a few things. So I think off the top of my head, there was some really significant differences in vitamin B production that we saw in the oat milk waste diet. And there were also a few other things as well. But those are things we sort of hone into. And because we have an isolate collection, our next steps are really then kind of putting them together and trying to create a probiotic, which we then would test. And so in this case, the goal is really to see if we could achieve those same growth differences with just one of these diets individually. So if we could just take the oat milk waste and apply a probiotic based on the findings from the combined diet, could we then achieve this growth rate? So that's kind of the pathway that we're taking now. And that's sort of what we would take for like a more maybe complex diet. For other challenges, if there's like a specific nutrient or something that you are trying to break down. So example, if you have a high cellulose diet, our approach would be maybe a little bit different, where we would just try to hone in on bacteria that we know have cellulases and that have that function. So if you kind of know a specific function, if you wanted to produce volatile fatty acids or something, you would hone in on strains that have that function and work around how to try and introduce them into the microbiome. In this case, it's a bit more of like a complex diet. We don't know exactly what function we want. And so that's why we're drilling down into the differences to try and find out what's different, what's the metabolism that is probably linked to that improved growth. And then, how do we build that microbiome for them?
Thank you. And then Stephen Cook has a question about the chicken feed diet. Is it broiler or layer, starter or finisher? So do you know what kind of chicken feed it might be?
Such a good question. I feel like it might be broiler, but I'm not 100%. Shows that I'm not the technician in the lab feeding them the diet. It's just one from the pet shop.
Yeah.
So I'm actually not sure. Yeah.
Yep.
I don't know if Olivia's on the line. She would know the answer.
If it's from the pet shop, it might be layer.
It could be layer.
Like layer hens or something.
Yeah, it's quite a complete diet in terms of its kind of vitamin and protein content.
Yeah.
Yeah, neither of these diets are deficient. Like we analyzed them. Neither of them are actually deficient. So we weren't like it's a bit different to other studies where we're feeding them something quite poor in nutrients. These are both kind of sufficient diets. And so that's why we're kind of surprised they did super well when we combined them.
Yeah. That's interesting. Maybe there's like some amino acid or something that was lacking in it that's specific for chickens or something that's found in when you combine it with other feeds.
Yeah, it's a bit hard with chicken feed because they don't have to actually disclose all the nutrients. They just say it has like a vitamin mineral mix. So we don't know for sure everything that's in it.
Yep, absolutely. Phoebe has asked, what percentage FCR, food conversion ratio, or other growth metric improvement range are you expecting to be able to achieve with an optimized microbiome?
Honestly, that we're kind of guided by industry on that. So we sort of work towards a target that we're given. So if we're told like a 2% increase or a 5% increase is what people are aiming for from their usual, then that's what we would be trying to aim for. So I mean, that's really the information we want to know from industry is what your goals are, what you want to achieve in terms of feed conversion ratios or other kind of metrics.
Yeah. And then we have a question, are most of the microbes in the gut BSFL anaerobes?
No, so there is definitely an anaerobic component. But no, there's also quite a lot that is aerobic as well. So we have an isolate collection of around 200 isolates, and majority of those are aerobic, just because that's the easiest for us to isolate. We do have anaerobes, and we are starting to get anaerobes, but I guess largely because they're environmentally acquired, they have to kind of be present in the environment and in compost or in food. So they do get oxygen exposure. So kind of at the very least, they have to at least kind of be microaerobic and a bit tolerant of oxygen. But yeah, we do see a lot of anaerobes. We do see a lot of Clostridium. We do see these anaerocolumnar anaerosporobacter. We do see anaerobes, and they do tend to have like really beneficial roles, particularly in kind of fermentation. But no, there's a lot of aerobic bacteria as well.
Yeah. I just had one question. Is more diversity always better for the gut microbiome, or is it, if you give them a big bunch of species to select from in their microbiome and they can choose whichever ones suit their diet, is that a better way of going about it than giving them a tailored suite of a narrower range of microbes for their gut, tailored for their diet?
Yeah, this is such a great question, and something we're really interested in. So one thing that happens is you do get variation in how a microbiome assembles. So if you give them a whole suite of microbes, the resulting microbiome could be different every time. And that's something we're interested in, like how long kind of does it take to eventually converge on a beneficial microbiome if you just give them everything, and also kind of how much, I guess, selection and fighting is there between microbes to establish. We haven't actually done that experiment yet. It's definitely on the cards for us. But yeah, certainly I think the kind of consensus is you do want some diversity in the microbiome. You want cross-feeding, you want that kind of metabolic richness. But yeah, they do have kind of some base functions that, as long as they're there, it could be fine. I guess, yeah, the theory is that if you have that kind of extra diversity and those extra kind of redundancies built in, you maybe get a bit more buffering. So you potentially, where you particularly have a heterogeneous input stream, you might start to get a little bit of buffering where you don't have as much variation in your output, because that microbiome is able to be a bit more adaptable. Again, that's something we're interested in and looking at. We haven't done that study yet, but we're quite interested in using particularly the modeling and microbial ecology principles for kind of building in what we kind of call like a reverse ecology microbiome, where you can use that microbiome to stabilize and buffer diverse inputs so that you get a more stable output. But that's like a very, very complex question in terms of sort of understanding all of those trophic interactions and how the microbes interact, as well as the host interactions. Certainly from like an intervention point of view, cost-wise, it's going to be cheaper to just give them a smaller amount of strains. Like you really do want kind of the minimum beneficial community, just because it's going to cost a lot more money to be able to kind of culture them up and build them up in a community. So yeah, that's maybe why you would do that rather than just chucking everything in. But yeah, you certainly can chuck everything in and see how it pans out as well, and it would probably also work.
If I can just jump in for a second, these questions, this whole thing is just absolutely fascinating, especially for someone like me on the periphery of all this. It's incredible. I think there's only a few slides left. Can I suggest that we just go through the slides that do remain and then just open and continue the conversation for anyone who has to drop off? They won't miss too much then.
No,
okay,
thanks. That's a good idea. I think we do just have one more study left, so that's easy. So this is the final study. So this is again like what we would call a top-down intervention, where we're starting with a homogenized gut microbiome. So it's an undefined microbiome. And this was Olivia's honors project, but she was specifically looking at targeting it for a function. And in this case, she was wanting to target cellulose degradation or growth of black soldier fly on a high cellulose diet. And so for this study, she had a look at initially sourcing different microbiomes. So she did a pre-screen where she was either looking for high cellulose degrading potential, but also making sure there were going to be some bacteria already associated with BSF that we know are beneficial. And so she screened using some of the different wild soldier flies, but also completely unrelated species, things like beetles, termites, and rotting wood, and homogenized those microbiomes and inoculated them into black soldier fly larvae, which she then fed to this very, it was a very controlled system for her honors because she was trying to look at the directed evolution. So it was an azenic system, and it was a very defined low nutrient, high cellulose diet. So again, in these little fighter trays, and you can see from the photos and also from the growth, there was a lot of variation in how they performed and what microbiomes appeared, which is good. That's what she was aiming for because she wanted as much kind of variation to be able to select from. And essentially for her directed evolution study, she had an initial microbiome, which was the black soldier fly microbiome, which then for a directed evolution, you would feed them on the diet. You would take the top performing, so in this case, the ones that grew the most on a cellulose diet, you would take their microbiome and you would passage that through into a new naive generation of black soldier fly. So the microbiome is being selected upon, not the actual black soldier fly larvae. But the other additional step she wanted to ask was around perturbation. So if you introduce external microbes, would that actually improve the directed evolution process? And the theory kind of says that, yes, you could actually introduce more variants. And again, that's kind of what we expect based on the variants we see here. But there could be some trade-offs, and that's what she was really keen to investigate. And so for her, the perturbation step was basically a mix of microbiomes from the top performing samples. So the very kind of top of all of these tests, she took that microbiome, mixed it all together, and that was her exogenous microbiome. And so for the directed evolution experiment, there was a first generation. And for that one, she did inoculate them with the endogenous microbiome, which is the black soldier fly microbiome, the exogenous microbiome. So that's that external one made up of, the dung beetles and some of the wild soldier flies. And then this is the perturbed endogenous, which is basically a combination of these two. So the black soldier fly and the external microbiome. And what we did see in this first generation was that the external microbiome, again, really kind of outperformed the black soldier fly microbiome in terms of how well the larvae were able to grow on this low nutrient, high cellulose diet. And so that's really positive because it does mean you can introduce microbes and get this phenotypic effect, which in this case was ability to grow on a high cellulose diet. Then for the directed evolution part, this is where she then passaged the microbiome and transferred it to a new set of neonates to basically see if that microbiome would then also perform well in the next generation. And we had some really interesting results for this one. So again, and that's in this graph here, the purple is this perturbed endogenous, the blue is the endogenous. So we didn't do the exogenous for this study in terms of the directed evolution. But what we saw was that perturbed endogenous, so the introduced, having the introduction of the external microbiome with the black soldier fly microbiome, again, they performed better in terms of their mass, their growth on the cellulose compared to the endogenous. But in terms of how heritable that was, it actually wasn't heritable at all compared to the endogenous microbiome, which was heritable. And even though that's not the greatest slope, that's actually fine for heritability. And so what that actually means is even though as a treatment, the perturbed endogenous did really well and consistently outperformed the endogenous, it wasn't that predictable. So it means if you were to take a microbiome that was top performing in generation 1 and then passed it to generation 2, it wouldn't necessarily be a top performing microbiome. Whereas with the endogenous, if you were to take a top performing microbiome, it's very likely it would also be top performing in generation 2. So that was quite an interesting finding the kind of ecological theory behind that is it takes a little while for a new microbiome to settle. And because these were quite different microbes, so they're coming in from quite different species, not just closely related soldier flies, it takes a little bit of time to settle. And again, you just get that variation in how microbiomes can assemble, and that can vary each time. And so the theory is if we were to do this for more generations and we let this microbiome settle, ideally we'd start to see that heritability and that predictability come back into it. But that's something we are obviously yet to test still. And Olivia is looking into the actual microbiome of this. So this was for her honours thesis, which she submitted, and she's now working on the publication for that, where she's done the sequencing and is actually digging into the specific microbes that are associated, particularly with the high growth. Again, to try and identify microbes that might add that benefit of cellulose degradation to the black soldier fly, with the aim of being able to design a more targeted probiotic that you could administer to black soldier fly to help them with a high cellulose diet. And in terms of, next steps for us, like I said, we're really kind of using this toolkit and understanding of the black soldier fly microbiome to be able to design a bit more targeted interventions. So we do have an isolate collection now of over 200 isolates, covering a whole lot of taxa across the board, which means we've just got that ability to build different microbiomes and test different things out depending on functions that we're interested in. And we do have, as I mentioned earlier, that really strong modeling capability. So Matt Morgan's team and also JP have been working to really develop some AIML tools and genome scale metabolic modeling so that we can actually do a lot of this in silico. It takes a lot of time and resources to be able to do all the testing to get different consortiums and different trials. And so we can essentially run this in a computer and try and hone in on what we think are going to be the most beneficial combinations for a specific output. And then we can take those, run them in the lab using our in vitro and in vivo screening systems, and see if they actually do validate what the models show. And obviously, if they don't, we kind of take it around in a cycle and learn and improve as we go. But that's essentially the capability that we're building at CSIRO. And I guess where we are now is in terms of kind of seeking industry engagement and research collaborations to target where we should go with that. We have a great toolkit. We know we can intervene and we can change the black soldier fly microbiome for specific outcomes. So I guess we're really interested in knowing, out of all of the types of things that you could do, what is most interesting to people and what is most interesting to industry, and where we should target our efforts next. And if people are interested in working with CSIRO, this is just a quick table to show there's a whole bunch of different mechanisms, from things like having a PhD student to the Kickstart program for small to medium enterprises, more to kind of just contract R&D or CRCP investments. So if people are interested and do want to do further work with us, I'm more than happy to chat as well later on. And again, thank you to AgriFutures, Emerging Industries, and to Steve and to Mo for hosting me, and to Alex for chairing the session tonight. And also a big shout out to everybody on this slide. It takes a community, much like a microbiome, to get this work happening. And we've also had really great support from industry in terms of giving us samples, giving us substrates to test, giving us funding as well, and collaborators at universities that have really helped us over the years. So happy to take questions now.
Great. Thank you so much, Amy. That was an absolutely awesome presentation. And there's so much potential in it. And really looking forward to, rolling it out through industry and seeing how it can help productivity. Phoebe has a statement. Thank you, Amy. From an industry perspective, this webinar has been spectacular. Can't wait for the next paper. And what do you need from industry, if anything, in the near term to support your work?
Yeah, that's thank you very much, and great question. So things that we would love to know from industry are specific targets. So are people interested in things like general waste bioconversion? Are there specific substrates people are interested in? Are there specific outputs? Do you want specific amino acids? The more specific kind of the easier it is for us to actually target to. But yeah, any kind of directional inputs into what would be most valuable would be great. Other things that are also really good for us are actually having waste streams and larval lines and things from industry where we can do the experiments as well. We can scale them down and do them in the lab using industry lines and feed. We've done that with GoTerra. They've been incredibly helpful, and that's been useful for us because our lab lines can be so different sometimes. They're quite good for a rapid screen. But when we want to actually make sure our lab findings are compatible with industry, it's always best to actually get actual waste streams and actual industry lines with their genetics and their microbiome intact, where we can see how applicable the interventions are and how generalizable they are. So yeah, those sorts of things are obviously really useful. And obviously, yeah, super happy to talk specific projects and co-investment opportunities as well, if there's like a specific challenge that people are wanting to address.
Thank you.
But yeah, send us the
Pujatu says, interesting insights on BSFL microbiome. How much of the microbiome is passed on through the generations? When would it stabilize if the diets keep changing?
Yeah, I mean, so back to maybe this study, the heritability is like 37%. So, I mean, this isn't a direct because they're going to naive generations, but assuming a similar kind of, if you had FRAS and you had kind of continuity, you would sort of see potentially this. If they're kind of kept entirely separately, they're different to other insects. There is no kind of direct vertical transmission from mother to offspring. So you kind of have to actually make that exposure happen. But with a variable diet, yeah, it doesn't really stabilize. It's kind of constantly changing. You might see certain beneficial taxa stabilize if they're kind of just generalizable and useful across different waste streams. And you might just see sort of more of the rare taxa is where you might see a bit more change happening potentially. But black soldier flies have a very like environmentally diet-driven microbiome, so is very flexible and very changeable. So the only way like for this study we get it to stabilize is obviously keeping them on that same diet and transferring that same microbiome over and over again, and eventually this would start to look a bit more like that, but hopefully with the higher function still.
Thank you. And David asks, how do these fields fit with the nutritional design of substrates, or is that a different range of studies altogether?
Sorry, can I get you to repeat that question?
How do these fields fit with the nutritional design of substrates, or is that a different range of studies altogether? I think this was referring to an earlier slide.
Yeah. I think, yeah, so talking more about the actual nutrition of the substrate versus the microbiome, they are very interacting. So I guess back to one of my very earlier slides where I talk about intervention and prebiotics, I mean, diet is part of that as well. So manipulating the diet is a way to manipulate the microbiome. So you can do things like actually add microbes, but you can change that microbiome just by also changing the diet nutrients as well. I don't know if that answers the question. I guess if you're asking more around like how do you kind of optimize nutrition and substrates, that's probably a little bit more separate. But yeah, just because diet and microbiome are so connected, and because the whole black soldier fly waste bioprocessing system is holistic, it's not just the flies on their own, like they're in the substrate, there's the frass being produced, they're reconsuming it, it's all kind of being mixed up. So it kind of is hard to separate the microbiome from the diet in that sense.
Yeah, that's a great answer. Thanks. I think the nutritional design field perhaps most commonly assumes that you've got a black box average microbiome in the larvae, and works in that respect. And actually, it seems to work out surprisingly well. So what that tells you is that with this extra level of complexity, there's going to be a massive range of extra possibilities if you do nutritional design and microbiome design.
I would agree with that. Yes. Yeah.
Thank you.
I have a similar question, sort of at the other end though, and is that can the microbiome affect the nutritional profile of the insect itself? So could you tailor the microbiome inoculant to get a different amino acid or fatty acid profile or more crude protein or whatever?
Yes. So yes, it does. It does. So similar, I guess, in the way that changing the diet will change the output, the microbiome will also have a role in changing the output just because you're changing the metabolism and the potential nutrients that are available to the host. So yeah, that's certainly possible, and that's certainly something we're interested in as well. So if you were trying to aim for high lysine or something in your black soldier fly, that's where a microbiome could be designed where you've got strains that are high producing lysine. I guess where the question is that maybe is a little bit murkier is even if you have higher lysine, what I guess are the limits of the black soldier fly and being able to then actually incorporate that and take that up. So yeah, there's definitely kind of a bit more to dig into there, but in principle, yes, you can change things like the lipid profile. You can change things like the amino acid profile, at least to a certain extent using the microbiome. And so that's something we're quite interested in as well.
And that's
you
even
can also
if it's
change
fed on
the frass too.
sorry. That's even if it's fed on the same diet. If you just
Yes.
if you keep the diet and
Yes.
change the microbiome, you can get a different
Yes.
nutritional profile of the insect. Yeah, very cool.
Yes.
That's very
Yeah.
interesting. I wonder if you can use microbiome, really tailor the microbiome to produce things that would otherwise never be produced by a black soldier fly. Specific enzymes or pharmaceuticals, perhaps.
Yeah, yes, definitely. I mean, that's something we are interested in as well. That's obviously like on the scale of challenging things, a lot more challenging, but it is definitely an avenue and something we're really interested in. So we do have a project funded around plastic degradation. And, as most people are aware, black soldier flies don't break down plastic. And so that's one thing we're looking at is can we actually design a microbiome that has that plastic degradation ability and introduce that into the soldier flies to kind of add that to them. And again, for that, we're really considering the whole substrate. So not just the gut microbiome, but the microbiome in the substrate as well. So you've got a bit of external breakdown of plastic as well as in the gut as well for plastic, just because that's such a difficult thing to break down. But we're certainly interested in that for other contaminants as well. So I know things like chicken manure are really high in larvicides, which kind of limits that as a substrate to feed to black soldier fly. And there are certainly bacteria that can break down and detoxify those kinds of pesticides. And so we're quite interested in could we use a microbiome and could you inoculate that in with your soldier flies? And would that then offer like a protection and enable them to be able to break down the pesticide in the diet that they're eating? So yeah, those are really interesting questions. They are a little bit more challenging because you are starting to add like a really novel and challenging function, but it's certainly not out of the realm of possibility. There's some other things too we looked at, was by accident, but we had a bacteria in one of our samples that started producing a pigment, so the astaxanthin pigment, which is quite important for aquaculture. They add like a synthetic one to try and make the salmon pink. And so we kind of had ideas around could we use the microbiome to produce this pigment so that if you fed black soldier fly, it would then produce the pink pigment and they wouldn't have to supplement that. So there's kinds of those ideas that we have. But yeah, they're definitely on the more challenging side, but certainly not impossible. And we're certainly interested in trying to tackle those too.
Yeah, that's so cool. Yeah, just a whole world of opportunities there.
Yeah.
Stephen Cook says, thanks very much, Amy. Good, cheers. Well, we'll follow up with further questions later.
Yeah, thank you.
And David has his hand up as well.
Can I ask one last question? I promise.
No
Amy,
problem.
some will Some will swear by using multi-generational selective breeding trials on a single property, most usually larvae weight or something like that. I wonder how you'd respond to that as an approach. I'm not quite sure, because I see some of your slides show very dynamic and unpredictable effects that are happening just through the right combination of microbiome, etc. How would you respond to that?
Yeah,
and, whether a single trait breeding program is wise or might be improved on?
Yeah, it's a good question. So yeah, going into kind of the host genetics and microbiome interaction, it's hard to answer. I guess what you kind of want to know is the trait that you're selecting on. You want to know how heritable that is to begin with. And this is where maybe other livestock have a lot more advanced knowledge in their kind of breeding trials in terms of like cattle and sheep. They know a lot more around specific genes that are linked to specific traits, which makes that very easy to select on. And I'm not sure if that exists for black soldier fly, but I'm not entirely over the kind of host genetics and pop gen and breeding genetics for black soldier fly. But what you need for that to work is a gene or a set of genes that are very, very heritable and that you can select on, that are always going to reliably give you that phenotype. And then you also need to know kind of what component of a phenotype is to do with host genetics and what component is actually to do with environment. And microbiome kind of sits in the middle because it is environmental, but the microbiome itself is, I guess, adding its genome to the host as well. So I guess both are kind of levers to pull on. And I think you can certainly get to a certain point with doing selective breeding, but you'll eventually kind of hit a limit. And I think the kind of the microbiome and other traits are additional levers that you can pull, and kind of seems a waste to ignore them. That's my opinion anyway.
Thank you. Great response.
I might suggest that perhaps we wrap it up here. There's been some fantastic
I'm going to stop sharing so I can
interest
see
and input, of course. If Alex and or Amy want to wrap anything up or any final comments?
No, I'm all good.
Just a massive thank you.
Just thank you to everyone, and more than happy for people to reach out if they have additional questions or just want to chat more.
Yeah, thank you so much, Amy. We'll do our best to make sure industry gets in touch with you and stays in clear communication with you, so we can make sure we're all in this together.
Yeah, excellent. Yeah, I mean, it's no point doing research if it's not really going to go anywhere. So yeah, always keen to make sure what we're doing is relevant.
Amazing. Well, thank you so much for the presentation, and thank you for your time this evening.
Thank you for having me.